Hello everyone, and welcome to Lesson 2 of our microbiology course. In this lecture, we're going to be talking about the history of the field of microbiology and some of the early developments in that field, what they led to, and how they still influence us today. On this slide, you have your lesson objectives. These are the things that you should be using to kind of guide your listening, guide your note-taking, things that you should be paying special attention to, things that you should know how to do or know how to describe, etc. by the end of watching this video and reading the chapter. In our last lecture, we talked about the importance of microbes and we talked about what microbes are. So we talked about those three different main categories of microbes as well as their subcategories. We talked about the prokaryotes, the eukaryotes, and the acellular microbes. And we talked a little bit about lenses and what they allow us to do. And now it's time to get into some of the early days of microbiology, how this field developed, and why it is still so significant today, and how even these early discoveries still are used on a daily basis by all of us, really. So in this slide, we're talking about the origin of microbiology. And the origin of microbiology really starts with the origin of lenses and the origin of microscopes. So while people may have had an idea that there were organisms too small to be seen, they couldn't confirm this, and they couldn't really understand the effect that these organisms might have on us and on our animals and on our crops without being actually able to see them. So microscopes were really key in the origin of this field because to be able to do anything in microbiology, at least in the early days, we had to be able to see these microbes, analyze them, identify them, and understand how they could affect us. Now, there are a lot more details than I'm going to go over here in terms of the development of lenses, but we're going to hit some of the highlights. One thing to keep in mind is that I will never test you on a date. To some people, the dates are really, really important. To me, I don't test you on the exact dates. It's a good idea for you to kind of understand general ideas in terms of the general timeline or time period, but I'm never going to have an exam question like, what was the year that Zacharias Janssen developed this lens? I don't find that useful. I don't think that is useful in your life, and I don't think it's going to be significant in working with your patients ever, most likely. So we're going to hit some of the highlights in the development of microscopes, then go over a little bit more about why they're so important, and move on from there. So early microscopes really depended on the development of lenses. And it was in the kind of middle to late 1500s that lenses really started getting good enough that we could use them not only in things like telescopes, but start turning them the other way as well and use them to magnify smaller things that were close to us. So the person that is credited with developing kind of the first microscope and the first lenses that were used in microscopes was named Zacharias Janssen. He developed these, or at least published his development, in the late 1500s, and he was able to create these lenses that could be used to magnify small objects. Over time, these lenses were modified even further, and they developed into what we would consider kind of some of the early or rudimentary microscopes. We've got an example of one of those right here. The first person that, again, we give credit to for using the microscope to look at cells is Robert Hooke. So in the kind of mid to late 1600s, Robert Hooke took these early microscopes that we had and used them to look at plants on a microscopic level. And he saw these little boxes. He saw that there were repeating units of little boxes that he called cells. We now know that those cells, those repeating units, are the basic unit of life or the individual units of life, and that when we put enough of them together, we tend to get these larger organisms, things like plants and people and animals. Now, a little bit later than that, we had another person named Antoine van Leeuwenhoek. He was a Dutch textile merchant. So he dealt with fabrics primarily, although he seemed to sell other things as well. So we'll just call him a Dutch merchant. And he was also kind of a scientist on the side, which was fairly common in those days. He took an early microscope, and this is kind of a reproduction of it, or an early example of what he was using to look at organisms with. And he decided to look at things like pond water, feces, tooth scrapings, and just kind of all kinds of little samples or other samples that he could get his hands on. And he, at least as far as we know, is the first person to actually observe microbes. So he is the first person, at least he's credited with being the first person to observe these microbes in the environment, in things like pond water, in fecal matter, in tooth scrapings, and in all kinds of other samples that he checked out. So he really is one of our key players in early microbiology. Of course, our other people matter too, and of course, like I said, we're not hitting every single important person, just going over the highlights here. But he was the first to observe microbes. So that's pretty significant. He published his work, and that spread all over Europe, and that allowed other people to start seeing microbes as well, understanding that they were there. And by being able to see them and see that when we saw maybe a rotting piece of fruit or something like that, that was actually microbes, we started to understand that maybe we could have some control over these. So what was really key in the development, or one of the key things that came out of the development of microscopes, is that not only could we now see microbes, we could see that they had a role in certain phenomenon in our lives that we may not have been able to understand before. So in terms of the importance of microscopes, one, like I said, just to reiterate, they let us know that microbes existed. We could definitively confirm that there were living organisms out there that we could not see, but that were around us everywhere. And they let us start to understand that these little living organisms could have an impact on our lives, that maybe they were involved in diseases, and maybe they were involved in food spoilage. And so a lot of research started taking off from there to understand how these microbes affected us. Around the same time that people were discovering microbes and starting to understand that they could play a role in our lives, and also as a result of this discovery, people started asking questions about the origin of living organisms, the origin and nature of disease, and developing a bunch of different advancements in medicine, healthcare, and food processing. So on the next several slides, we're going to talk about how the early days of microbiology and our early microbiologists helped people understand that spontaneous generation, this idea that organisms could just kind of pop out of nowhere, was incorrect, how they helped develop cell theory, how they helped understand the origin and nature of disease through what we call germ theory and Koch's postulates, as well as talk about these advancements that came about, things like hand washing, sterile surgery, disease prevention, disease treatment, and really get into the context of those in that time period in history. During the time when microscopes were being developed and microbes were being discovered, one of the major ideas that was held about the origin of living things was called spontaneous generation. And spontaneous generation basically is this idea that modern life forms, or life forms as we know them, arose or could arise from non-living components. So the evidence that people thought they had at the time for this being the way that life arose was that oftentimes they would see, they would have these large piles of grain in their property, and they would see rodents. They would see mice or rats kind of coming out of those piles of grain. And so they looked at that and said, oh, hey, maybe that pile of grain is like the parent of these mice, that pile of grain creates the right conditions for mice to just show up. They often would see maggots coming out of meat. So if they had some rotting meat and maggots started crawling out of it, to them, that was evidence that, hey, this meat gives rise to these maggots. Or they would see mushrooms coming out of wood or piles of leaves or something like that, and they would say, oh, wood or leaves must be, the parent of these mushrooms. And so, while not necessarily everyone maybe thought this was one of the big ideas about certain types of life forms, that they could just show up. They could just kind of pop out of nowhere as long as these environmental conditions were right. There were people who questioned this, though, and there were two people who were influential in disproving this definitively. So we're going to talk about them on the next two slides. But basically, Francesco Redi was able to disprove this idea of spontaneous generation for macroorganisms, and Louis Pasteur was able to disprove the idea of spontaneous generation for microorganisms. Francesco Redi was an Italian physician, naturalist, scientist, etc., who was one of these people who questioned this idea of spontaneous generation. He said, I'm not entirely sure that maggots just arise from meat or that mice just arise from grains. I think that maybe they need parents the same way that, humans need parents, or dogs need parents, or cats need parents. And so he developed a really elegant experiment involving the use of meat in jars, and was able to disprove this idea of spontaneous generation for macroorganisms. And macroorganisms, just to define that, are organisms that we can see with the naked eye, or organisms that we can see without help of a lens. So Francesco Redi was able to disprove this idea of spontaneous generation of macroorganisms, which means he was able to disprove the idea that, dogs or mice or humans or trees could just pop out of nowhere. So he took these 3 jars of meat and he covered one completely. So basically he sealed it completely with a lid. With another one, he basically took some gauze or some cheesecloth, whatever you want to call it, and put it over the top. So I'm just drawing the gauze there because there wasn't a great image for that. So he put that over the top, and that allowed air to get in. It allowed things like flies to know that that meat was there. And then he took one jar and put the same type of meat in there and left it completely uncovered. He let this sit for a certain amount of time, a couple days, for a week. So I'm just going to write out time right here. And he observed that over that time period. And he noticed this. What he noticed was that the meat that was completely sealed off didn't attract any flies and didn't grow any maggots. So no flies, no maggots. The jar that had gauze on it attracted flies, and maggots did develop, but not on the meat. They developed on the gauze. So flies and maggots on gauze. The jar that was left completely uncovered attracted flies, and there were maggots on the meat. Sorry, that's a little messy. I'm running out of room on the screen. So, recap. The jar with meat that was completely sealed, completely covered, had no flies and no maggots. The jar that was covered with gauze had flies that were attracted to it and had maggots on the gauze. And the jar that was left completely unsealed had flies that were touching the meat, that were totally attracted to the meat. And then maggots developed out of that meat. And so what this tells us is that maggots do not just spontaneously generate from meat. Because if they did, then these 2 pieces of meat should also have maggots on them. But they don't. What this tells us is that the fact that we can see that there are flies and maggots on the gauze and there are flies and maggots on the meat in this jar, tells us that maggots have to come from flies. If there are no flies, like in the completely sealed jar, there are no maggots. And so this idea that, maggots could just spontaneously generate out of meat was false. That is not true. And this experiment showed us that. It showed us that macroorganisms, larger organisms, must have parents. They must come from other living things. They can't just spontaneously generate. And so that discovery helped, push people into a new direction in terms of understanding where living organisms came from. But there was still kind of a lingering question about whether microorganisms could spontaneously generate. So there was, this disproved the idea that macroorganisms could, and people could accept that. They accepted this experiment and said, okay, that makes sense. But there were still questions about whether microbes could spontaneously generate. They thought that maybe these microbes that were so small that we couldn't even see, maybe they just popped out of nowhere. Fortunately, we had someone else come along and say, I don't think that's true about microorganisms. I don't think they can spontaneously generate. So Louis Pasteur is one of the formative figures in microbiology. He is one of the first major microbiologists. Some people call him the father of modern microbiology. And he did an experiment that definitively proved that microbes did not spontaneously generate either. To do this experiment, he took 2 flasks, and these flasks, the shape of them is really important. So these are called swan neck flasks. And they were significant in their design because they had kind of like a curved straw that basically allowed air to travel in and out of the flask. But in this curve here, they would trap any heavy particles that were in the air. So they would trap dust, they would trap pollen spores, they would trap any hairs or just any large particles that happen to be in the air. And what he did was take 2 of these flasks. They were identical flasks. He filled them with broth, and then he boiled that broth. So people did know at the time that organisms need air to live, so he made sure that there was air present. And they also knew that if you boiled something, it killed things. Things didn't grow. Like they knew that heat was a way of killing things. So he took 2 flasks, identical in every way, filled them with broth, and he boiled them. And for one of them, he left that swan neck intact. He just left it as it was, just left it sitting there. The other one, he broke off that swan neck. So he took this one, he broke off this swan neck, and basically left it open to the air. So in this flask up top, the air could get in and out, but anything that was in the air would get trapped right here in this little bend. This one was just open to anything in the air being able to fall in. He let those sit over time, so gave them some time. And he observed them as that was going on. He noticed that the broth in the flask that still had its swan neck on stayed completely clear. Nothing was growing in it. But he noticed that in the broth that had in the flask that had its neck broken off, it turned turbid, meaning it turned cloudy, and things were growing in it. Sorry, it's cloudy, not clear. So he noticed that the broth that had been open to the air, basically exposed to the air, was cloudy. and that the broth that had been exposed to the air but had its swan neck intact and anything in the air got trapped remained clear. And he could observe these things under the microscope, and he could see microbes in this broth and no microbes in the top broth. And so this tells us that microbes do not just spontaneously generate, because if they did, we would have seen microbes in this broth as well. But we didn't. We only saw microbes, or he only saw microbes, in the broth that had been exposed to all of the particles in the air and things had been able to fall into. So this definitively proved that no life forms, whether they're macroorganisms or microorganisms, can just spontaneously generate. They have to come from somewhere. Some other living organism has to reproduce and give rise to other living organisms, including microbes. As a result of spontaneous generation being definitively disproven for both macro and microorganisms, a new theory arose. So biogenesis replaced the idea of spontaneous generation. And biogenesis states that living organisms arise from other living organisms. Basically, life arises from life. It doesn't just pop out of nowhere. It doesn't spontaneously generate. That if we have a living organism, we know that it came from another living organism. And we know that that living organism has the potential to give rise to other living organisms. Now, at the same time, or around the same time roughly, that biogenesis was developed as a theory, cell theory was also developed. Now, cell theory states that all living things are made of cells. Cells are the smallest or individual units of life, and reproduction makes similar cell or organism types. So basically what that says is cells give rise to like cells or similar cells, and organisms give rise to like organisms. So if you have in the human body a liver cell and it reproduces, it's going to create a liver cell. It's not going to randomly create a skin cell. It's not going to randomly create a blood cell. It's not going to create a neuron. It's going to create a cell like itself. And organisms as a whole are going to do the same thing. So a dog isn't going to reproduce and create a tree. A cat isn't going to reproduce and create a giraffe. A whale is not going to reproduce and create a mushroom, right? A dog is going to have puppies. A cat is going to have kittens. A whale is going to have a baby whale. So, and plants are going to have plants. They're not going to spontaneously create like a dog or an elephant or something. That organisms and cells are going to give rise to things that are similar to themselves, not just randomly reproduce and create something entirely new. And these ideas really were key in us understanding some very important things about microbes and the nature of disease. So we're going to talk on the next several slides about how important the idea of biogenesis and cell theory were to our understanding, our modern understanding of what can cause disease. For much of human history, people didn't really know what caused disease. There were lots of different ideas in lots of different cultures spread throughout the world. But some of the popular ones were that you breathed in bad air and that made you sick. You ran into spirits and that made you sick. Or you were being punished. It was some kind of divine punishment because you were bad and this was the result of your bad actions. Fortunately, there were people that thought that maybe there was a different cause of disease, and they started looking at these microbes that had been relatively recently discovered and considering things like biogenesis and cell theory and trying to think about how all of those things could fit together. So one of the new ideas about disease was called germ theory. Germ theory is the idea that microbes can be, and sometimes are, the cause of disease. That diseases are not necessarily caused by spirits or bad air or divine punishment, but instead it's because you came in contact with this microbe, the microbe got into your body and caused damage, and you got sick as a result. So this idea of germ theory was put out there, and many people studied it and had a lot of evidence to back it up. And there was one scientist named Robert Koch that really was instrumental in helping us kind of solidify this idea of germ theory and providing this kind of definitive evidence that many diseases were indeed caused by microbes rather than things like spirits or bad air or divine punishment. Robert Koch did a series of experiments where he took diseased and non-diseased animals and basically kind of passed microbes from diseased animals to non-diseased animals to see if that microbe caused the same types of illness in that non-diseased animal that he was using as the previous organism had had. So I'll explain that a little bit more in just a second, but basically that was just kind of the big picture of that. Koch's postulates are a series of experimental steps or a series of clinical requirements that help us understand how certain microbes relate to certain diseases. So these are steps that help us definitively link specific microbes to specific diseases. And the basic assumption or the basic premise behind all this is that each time we see a certain disease, then there should be a certain microbe present. And each time we see a certain microbe, we should be able to see that same disease. There are several steps for testing this. First, what we do is isolate what we think the disease-causing agent is. So we take a sick or dying animal and we take a sample from it. We take that sample from the animal and put it into a place where that microbe could grow. We grow that microbe up and we look at it under the microscope. We identify it, we look at its characteristics, we maybe look at if it's moving, how it's moving. Basically, we just gather as much information as we can about this microbe that we think caused the disease in this animal. Then, once we have enough of that microbe and we've studied it enough, we can take that organism and put it into a healthy animal. After that, we observe the healthy animal, and if that healthy animal gets sick, then what we will do is take a sample from that sick animal, put it into a place where that microbe can grow. We look at it under the microscope again, and we look to see if that microbe from the first animal is present in the second animal. We look to see if that microbe matches the microbe from the first animal, if it has the same characteristics, the same shape, if it moves the same way. Basically, we just check to see if the organism that made this animal sick, animal number 2 sick, is the same organism that made animal number 1 sick. And so our steps for testing is that we have to isolate the organism from a diseased or dead animal and culture it and study it. Then we take that organism and check to see if it causes disease in a healthy subject. And then we take that previously healthy subject that is now diseased and we check to see if that organism that made animal number 2 sick matches the organism from animal number 1. And if it does, we can basically say that this organism is the cause of that disease, not a spirit that the animal came in contact with, not divine punishment, not bad air. Instead, we're scientifically evaluating whether an organism, whether a microbial organism caused disease in some kind of animal subject. And so Koch's postulates, the series of experiments he did, helped us understand that microbes were oftentimes a cause of disease, and that certain microbes cause certain diseases. It wasn't just that microbes in general caused every single, like any general microbe could cause any general symptom. It was that microbe A caused disease A and microbe B caused disease B. It wasn't that microbe A would cause disease B under certain circumstances and disease A under other circumstances. No, that wasn't it. It was that microbe A always caused disease A. Microbe B always caused disease B. Microbe C caused disease C. So we were able to link not only this idea that microbes could cause disease, but that specific microbes could cause specific diseases. And this was huge on several levels, because what this showed us was that diseases had natural origins, basically. They had origins from microbes, in many cases at least. And if diseases came from living organisms, and living organisms came from other living organisms, then we could potentially have a way to control disease. We could maybe have a way to treat a disease, because if disease came from a living organism, we might be able to kill it. And we might be able to have a way to prevent disease, because if disease comes from a microbe and a microbe can't just spontaneously generate, then if we can cut off the source of microbes, we can potentially cut off the source of disease. As a result of this new understanding of microbes and their role in disease, as well as things like cell theory, biogenesis, germ theory, Koch's postulates, all of those things led to several developments in medicine and healthcare, as well as in what I called food hygiene or food practices. So because people started to understand that many diseases were caused by living organisms, by these microbes, and because they were living organisms, they could be killed, or if we could cut off the source of them, we could prevent the disease entirely. Lots of scientists and physicians started to really reevaluate a lot of very common medical and healthcare practices. So one of the main things that came out of things like germ theory and Koch's postulates was the idea of hand washing between patients. Prior to things like germ theory, Koch's postulates, etc., hand washing between patients wasn't necessarily something physicians did or nurses did. They might kind of, if they were really dirty, they might kind of wipe their hands off on a piece of cloth. Maybe they washed them sometimes, but definitely not always. And it wasn't until a Hungarian physician/scientist named Ignaz Semmelweis came around and realized that hand washing between patients might actually help prevent diseases. He noticed that in labor and delivery wards, basically if you had several women kind of lined up in a series of beds, we'll say woman A, B, C, D, E. If woman B got sick after childbirth and the physician went from her and treated her and then went to woman C would often get sick as well, and so would woman D, and so would woman E, whereas woman A tended not to get sick. So he realized that disease seemed to be being passed kind of in a line from woman to woman. And the thing that was common between all these women was that the physician had touched each of them and probably not washed his hands between them. So he started this, at the time, novel idea of washing his hands between patients. And he noticed that if he did that, even if you had these same women, women A, B, C, D, and E, if woman B got sick, but he washed his hands in between woman B and woman C, then woman C didn't get sick. And if he washed his hands between woman C and woman D also didn't get sick. And so he realized that by washing hands, he was basically cutting off the source of infection between these women or between these patients. And he thought that was a great idea, and he published his studies, and most people did not really respond to that very well. They didn't like that idea. They thought it was kind of ridiculous. And so unfortunately for him, he was kind of ostracized a little bit until his work was accepted later on and until the importance of hand washing was really recognized. And now one of we know now that one of the most important things we can do in terms of cutting down disease of patients in hospital settings is for healthcare workers to wash their hands frequently. Another idea that was developed as a result of all of these early discoveries in microbiology was sterile surgery. So prior to, again, the understanding of things like germ theory and microbes and everything, people performed surgery, or what we'd at least consider rudimentary surgery, oftentimes without necessarily washing their hands or using clean instruments or having a clean, sterile field to work in. But we had a scientist named Joseph Lister who thought, maybe if I clean the area and I clean the instruments that I'm working on, we can help prevent disease in these surgical sites. So Lister came up with this idea of cleaning instruments between patients and before the first patient, obviously, and having the people who are participating in surgery have clean hands and having a sterile field around the opening of the body that the physician or the surgeon had made. So he used different gases to help clean the instruments and create a sterile area around the part of the body that was being operated on to help prevent microbes from getting introduced into that surgical wound. People also started thinking that maybe they could treat diseases, again, since these were living organisms and living organisms could be killed. Maybe if someone got sick, we could give them something to help kill those organisms that were in their body. And so antibiotics were one of the major developments as a result of the understanding of the nature of disease. You're looking at a bunch of different antibiotics and pills right here. We talked a little bit about antibiotics in the first lecture and Alexander Fleming and how he discovered that certain chemicals that bacteria and fungi would produce would kill other bacteria. And so antibiotics were one of the major discoveries or major developments as a result of things like germ theory. Sorry, it's a little messy. And then another big idea was this idea that maybe we could prevent diseases. If diseases are caused by living organisms, maybe we can cut off the source of disease and prevent them. And so vaccines, there was an idea that maybe we could help someone's immune system be ready to fight off a disease before the initial time that they got exposed to it. So vaccines are products that are designed to give someone a small amount of a dangerous microbe. Basically to help prepare their immune system. To help prepare their immune system so that when they get exposed to a large amount of that microbe in a real life setting, their immune system is ready. And the person we credit for modern day vaccines is named Edward Jenner. I'll just put his name here. This is a parentheses, not like a weird G. And he kind of got this idea that we might be able to expose someone to a small amount of a microbe to protect them from a bigger disease later, when he noticed that women who milked cows oftentimes got a disease called cowpox. And that cowpox caused lots of lesions, similar to what you would consider chickenpox today, on their arms, but never really spread anywhere else in the body. He noticed that those women who got cowpox didn't tend to get smallpox. And so what he did was he took this boy and he developed this solution built off of cowpox, gave it to this boy. So he took this, what was the early, kind of an early type of vaccine, gave it to this boy, and then he checked to see if he exposed that boy to smallpox, whether he would get it or not. Fortunately for this little boy, he did not get smallpox, and Jenner started making larger batches of this vaccine and distributing it to other places in Europe at the time. And smallpox, as a result, is the first disease that people have been able to completely eradicate, which means wipe out. So smallpox was the first disease, and really the only disease so far, that has been able to be completely eradicated or wiped out as a result of vaccination efforts. So after the smallpox vaccine got going, people started developing an interest in preventing all kinds of other diseases, and vaccines for things like anthrax and rabies, as well as things for diphtheria, pertussis, pneumonia, meningitis, etc., have been developed as a result of this initial idea about vaccines. So those were some of the significant developments in medical and healthcare that came about as a result of our new understanding of microbes and disease. And just a little bit about like what I call food hygiene developments. People started getting better with refrigeration and heating. They realized if food was kept cold or cold enough, then microbes did not grow in it. Or if it was kept hot enough, then microbes did not grow in it. A process called pasteurization was created as well. So pasteurization is named after Louis Pasteur. He's the one who developed it. And basically, this idea of pasteurization is that you heat up liquids to a very high temperature for a very short amount of time. And this generally maintains the property of the liquid. So it maintains its taste, it maintains nutrient value, it maintains texture, etc. And then you cool it down very quickly. And that heating process kills not all the microbes, but a significant number of microbes in there. And then that very quick cooling process prevents them from being able to reproduce. So you create a liquid that may have some microbes left in it, but there are so few of them in there that it doesn't make people sick anymore. So things like milk and juice, etc., these are pasteurized. Basically, they are heated to a very high temperature very quickly. That kills off a significant number of microbes, and then they're cooled very quickly to help prevent any microbes that are left from growing again. So in pasteurization, we don't completely wipe out all of the microbes, but we get the levels of microbes down so that they're low enough they don't make people sick. And then improvements in canning were made as well. So people realized you could can food, and canning food basically deprives the microbes of any oxygen in there. And so that prevents them from being able to grow, but it maintains the food in a state that is safe to eat for months after, or potentially years after as well. So all of those things were developed, or in some cases at least improved upon, as a result of the understandings of how microbes could cause disease in the human body.